doing. Okay. Minimize this. Bear with me. All right. So again, this is just using this SSH command, which creates a secure, the first S shell, connecting into this server that's in a basement on campus. And once you provide your password, you should have the prompt, as we learned about last time. Yeah, question? No? Okay. All right. Anyone, please speak up if you're not at this point, if you haven't been able to connect. I'm not able to have my terminal open, the slides, and look at everybody, so I'm going to have to depend upon people just speaking up. Okay, so I'm going to assume that everyone is logged into Malibu, which is fantastic. What we're going to do now, the first thing we're going to do is learn on the command line how to do what we do often on the internet. You use Chrome or Firefox or whatever, and you have some video or a GIF or a picture that you want to download. You right click on it and you say save as, and it downloads it to your computer. Well, we can do the same thing if we have a URL on the UNIX command prompt, and there's two commands for that. One is called wget, and in the homework that I'll assign at the end of today, there's another one called curl that you'll learn about in the homework. Wget is the easiest. It allows you to download or get files if you give it the full URL to that file. So what I want you to do, first of all, I've posted the slides to the chat for Zoom today. And what you should be able to do is just double click and select this whole line and paste it into the terminal. Before I move on, I just also want to mention, if you have any problems with commands not working or anything like that, also feel free to interrupt me, but ideally connect to Sarah Lucas, Gage Black, or John Chamberlain, the TAs in the course via, just use the private message feature in Zoom, and they can probably help you out. All right, so we're going to download this file by copying and pasting this full command. And what happens is, this is a very tiny file, so it downloads it very, very quickly. Right. So now we should have a file. We can use the ls command to confirm, and there's a file called FASTA text, and you can do a full ls on the whole directory, and there it is. So you should have this file in your directory at this point. Any problems at this point? Please speak up if so. Otherwise, I'm going to move forward. All right, so moving forward for the rest of the course, we're gonna have example commands that are either illustrating what a particular command is doing, or I'm asking you to copy and paste that command or type it into the prompt. And I'm going to add a convention for all the commands where the beginning of the command starts with a $sign. That is meant to reflect the prompt. You don't copy and paste that. You're just copying and pasting the command that is after the prompt. Okay? So we learned last time about this command called head. And as I mentioned, head reports the first, by default, 10 lines of a text file. As I showed last time, you can give it an option -n and tell it how many lines you want. If you don't want 5, if you want, or 10, if you want 5 or 9 or 1 or 1,000 or 1,000,000, you just give it this number after the -n option. So let me show that again. So we've got this frost.txt file. And as I showed you last time, it just reports the first 10 lines of this poem from Robert Frost. I think one of the things that you should understand that you may not know yet is, I mean, how does it know what a line is and what a line is not? When we're scrolling through a Word document that we've written, we can see the lines, but what Unix really needs, how Unix distinguishes lines is at the end of every true line, if you've hit the return or the enter key, that introduces this hidden character called a /n. That is a new line character. So when you hit enter, what's sent through your keyboard to your computer is an instruction to add a hidden but actually present character in the file, which is a /n. So when this head command opens frost.txt, and you've told it you want the first 10 lines, it is starting at the first byte in the file and scanning until it finds a new line. When it finds that new line, it says, oh, that's the first line. And the new line goes to the start of the second line, keeps scanning until it finds that hidden character. You can't see it, but there's ways that I'll show you in a minute to see that character. But essentially behind the scenes, these programs, and this becomes relevant to many Unix commands, are looking for hidden characters like tab characters or new line characters like this. So essentially, when you say head frost.txt and it reports the first 10 lines, it knows when to stop when it encounters the 10th new line character. Okay? All right. So again, I can say things like -5, and in this case, it's finding the first 5 new line characters. There's a convenient command that I think you guys should know called, let me just move this bar here, called clear. It'll make your prompt, it'll make your shell erase all the previous commands, so you kind of have a clean desktop, I guess. So see this again. Right, so now we get the first 5 lines, all right? And again, even though the default is 10, you can specify I want exactly 10 or 11, all right? Pretty basic command, but one question you may have, and it's probably a valid question, is, why would I ever use this in my own research? Well, a lot of times we're dealing with big data files, and we might just want to peek at the contents to get a sense, well, does this file look like it's formatted correctly? Are there any obvious problems? All right. So I mentioned, we kind of know this already. I talked about it last time. If the head command looks at the first few lines, what do you think the tail command does? Anyone have a guess?
It looks at the last 5 lines, or however many lines you specify.
Exactly, yeah. So there's a question on chat. Okay, so audio is freezing up. I was so worried about this. I think what's happening is the VPN slows down my bandwidth, unfortunately. I have Google Fiber at home, but I think once I log in, I think some of you are encountering this as well, the VPN throttles things. All right, well, henceforth, I will go into my office in hopes of having better bandwidth and by not having to connect to the VPN. I apologize. Maybe what I will do is turn off my video so that I have a little extra bandwidth for audio. All right. Thanks for letting me know that. Please hit the chat remind me in the chat if it's still breaking up, and if it's getting really bad, we might have to table things. I didn't want to go into the office for obvious reasons, but if it's not going to work, then it's not going to work, and I'll have to make changes. All right, so we just used the wget file to download this poem that we've all probably heard during childhood. Now let's transition into downloading a file that is actually relevant to something we do in genomics. And what I want to start with is just downloading this little snippet of what's called a FASTA file. And if you're familiar, a FASTA file is often the file format used to store the nucleotide contents of a genome, say the mouse genome or the human genome, okay? So again, we're gonna copy and paste this whole command. As a refresher, wget is downloading this file called DNA FASTA at this URL, which just happens to be my home directory on the Center for High Performance Computing. If you don't know already, every user, when you create a CHPC account, you can create a directory under your home called public_HTML. Any file that you put in that directory will be available at the following URL, where the only difference is you just need to put your unit there. So it's a nice way to host a personal website or data files you want to share with other people, okay? In this case, I've just posted this little file for our purposes today. All right, and I already downloaded it. Before I move on, can you just confirm, is the audio a little bit better? Am I breaking up still? I couldn't hear that. Yes?
Yes.
Oh, right, but I asked two questions. Yes, it's breaking up still, or yes, it's better?
Yes, it's better.
Okay, wonderful. Thank you, Abby. All right. Sorry, let me fix this again. Use clear again to make this a little more legible, and download this file as you've probably already done. Right, so now we're going to learn a new command called cat. So head and tail look at the first few lines or the last few lines. Cat is essentially, it vomits the entire contents of the file to the screen. Okay, so it is opening the full thing. Looks like we got some questions here. Better, awesome, thank you. So cat, just do it. It spits out something you're probably familiar with, a lot of nucleotides. And it turns out that this is the complete nucleotide sequence for the human mitochondria. So this is going to be, what is it? Something like, should be like 16,000-ish, 16,500 nucleotides or something like that. Okay, and what's the key difference here is that, unlike looking at the first or last few lines, this is literally just opens up the file, finds the first byte, reports it all the way to the last byte. Once it sees that last byte, it shuts down and we're done, okay? But the cat command is really nice for obviously showing the contents of a file, but working in is some other cool features that the cat command has. One thing we can do is use the -n, and this has some parallels to the head and tail -n. The -n option in head and tail gives you the first n number of lines. The analogy here is cat -n is reporting the line number for each line. Okay, so we can tell now that there's 238 lines in this FASTA file, and each time it's incrementing that counter. When it encounters this, there's a little hidden /n, hidden new line character at the end of each of these lines, and that's how it knows the program, the algorithm within there is keeping a counter. Every time it sees a /n, it increments this counter, and that's what's reported when you ask for the -n option. Okay. Right. So now we can actually prove to ourselves that there is a new line character at the end of these lines. So the cat-e option reports a $sign to represent that hidden character. And the reason it chooses a $sign is that's a visible character. The /n, it exists in the file. It occupies, each of those /n's consumes a byte in the file. So if there's 238 lines in this file, the file is 238 bytes bigger than you might think it is because of that hidden new line character. And all this is doing is showing that those new line characters actually exist, and you can see them at the end. But this, so the question you're probably naturally asking is, why the heck would I want this? Who cares? Well, it's a great way to debug. Sometimes you get files from collaborators or downloaded from websites which are malformed, where they, a common occurrence, a very common occurrence, unfortunately, is researchers will share a file that was created on a Windows machine and then post it on the internet. And then when you download it to your computer, a UNIX machine, Unix and Windows use different new line characters. Fun. And so what happens is hilarity ensues when you download a Windows file and try to use it on Unix, because it's a different new line character, and programs like cat and grep and all these commands that we'll learn don't work as expected because it isn't the proper new line character. And so using this cat-e command on this malformed file, if something's going wrong, you can first check, well, are there actual Unix new line characters at the end of the file? If you see something other than a $sign, just a $sign, you know that something's wonky. So it's a good way to sanity check or figure out what's wrong with a file. Okay, so that's the real world application. Any questions about cat? It's kind of straightforward, but happy to answer any questions at this point. All right, great. Next command is WC, not water closet. This stands for word count. And this reports, by default, it reports three things, three very useful things. It reports how many lines, words, and bytes are in the DNA in this file. So if I do WC on DNA.FASTA, you can either type it or you can copy and paste from these little examples over here. I see, well, I've got a much bigger file than I had last year. That's okay. But there's 238 lines. Well, that matches up with what we saw with cat-n, right? This is one rule in science is get the same result two different ways. So we got the result that way. We got the result that way. 238 matches 238. I trust this result. The other thing is that it's got 243 words. That's a more complicated sort of computer science notion, but you could think about it as the blocks of texts. There's this block of text, and then there's, at the very beginning, there's these other blocks of text. And then lastly, it says that there's 16,800, said my internet connection was unstable, so I'll start again. It says that there's 16,863 bytes in this file. Okay, I've got a trick question for you. Can you guys hear me? Sarah tells me my Zoom is, or my audio is paused.
We can hear you now.
Okay. Boy, this is unfortunate. There are 16,863 bytes in the file. Is that how many nucleotides there are? Any guesses?
I think there's a header line, so it's not going to be exactly the same.
Okay, yep, that's one reason. So we certainly would have to take out the number of bytes in the header line. And then what else inflates that number?
Is it counting the new line characters?
Exactly. So the new line characters are part of the count as well. So we would have to take some steps to actually count the number of nucleotides in the file, just using something like WC. It's a little more complicated because, as was mentioned, sorry, I didn't catch the names as they popped up, but headers and new lines inflate that number, the number of bytes above and beyond the actual number of nucleotides. All right, nice job. Let's see here. So I'm going to take a quick tangent here before we move on to other commands to just talk about the notion of using tricks that exist on the command line to save you time. You've probably already watched, and I've told you that I'm a bad typer and I'm lazy, so I try to use as many shortcuts as I can. The first shortcut I want to tell you about is just using your arrow keys. If you hit the up arrow and keep hitting it, I can scroll through all the previous commands that I've typed. And one of the things that I think you'll find, especially when you're trying to figure something out, you end up trying the same command over and over again. So don't type it over and over again. Increase the rapidity with which you go through an experiment, iterate through an experiment by just using shortcuts like scrolling through. So if I wanted to go back and I don't know, let's find one that's actually useful. Look at the first few lines of the Frost file. I can do that. I don't have to type anything. I can just scroll through. That's one. The other one is this really nice command, I'll use clear again, called history. And not surprisingly, it gives you a history of all the commands that you've typed. So a couple cool things here. If I use history and I say, oh, this command, there's a command number with it. I can do exclamation point 744, and it'll run that same command again. So history, it knows, oh, you want to run the 744th command that you've executed in your history. Great. I don't always use this, but it's sometimes nice if you know, you just do this quickly and you don't want to copy and paste the whole thing. You see the number, and if you're a good typer, you can just knock out the number really quickly. I talked about the tab complete yesterday. So if you type LS and D, I can hit pound on the tab key, bash it a few times, and it'll give me my options. And obviously, like autocomplete on your phone, it sort of guesses based upon you know, how unique the combination of characters that you've already typed is, right? So that's one. And then the other one is this, I use this all the time, Control R. So here's the way it works. I'm just going to show an example. I'm on my keyboard typing Control, holding it down, and then typing R. And what that says is the R is for reverse search. So what's reverse search? It's really just allowing you to type in characters, and in real time, it's going to find, just like autocomplete, it's going to find commands that match the character that you've typed. So I typed LS, and at this point I can hit tab a bunch of times. Oops, sorry. It's halted. There we go. So Control R, and I can, you know, it'll find matches, commands that match the characters that I've typed. And then you can use the up command to cycle through the matches. Okay, the last thing I want to tell you, and this is something that comes up quite a bit. Anytime you've got a command that is running for too long, or you've realized that it's running and you did something wrong, you can always kill that command. And the way to kill it is to hold the control key, and then C. Okay, so let me see if I can give an example. Well, let's see. So sort, if I type sort, it's waiting for me to give it input. It's just sitting there. If I want to get my prompt back, I'll just do Control C and it'll give me my prompt back. Okay. All right, so now let's move into doing something that's actually useful, arguably, and more relevant to science. So let's imagine some of you are first year graduate students, the more senior graduate students and postdocs among us, imagine you're back to first year. And you work in a lab that studies gene regulation, in particular in the context of human muscle development. Well, as a grad student, as you're starting to collect data files that are relevant to your research, one of the first things you'll need, obviously, if you're studying gene regulation, is a list of all the genes and isoforms or transcripts that are present in the human genome. Oops, let me just fix this file here. And there are many ways to get these files. I've already created one for you, but we'll talk later in the course about resources like the UCSC Genome Browser and Ensemble. But these gene annotations or transcript annotations are most commonly represented in this file format called GTF, or gene, I forget what the T is. Is it gene transcript format? I can't remember what the T is, but let's just think G something format, gene something format. So what we're going to do right now is just create a new directory. So I mentioned this at the end of class last time. So if you copy this command, this make mkdir, make directory, oops, and type muscle development or muscle dev. If you ls, now we've got a new directory. Yours is going to be a little simpler here. You probably just have a few directories. So now we have a new directory, and that's denoted by this blue font. If I see, and then I can cd or move into, change into that directory, and there's nothing in there. Okay. So what we're going to do is download this genes.gtf file using the wget command again. Okay. It's a big file. It's 1.15 gigabytes. It took a little while to download, or like a second or two. Hopefully that should go as quickly for you, because this is, remember, this is all not being downloaded to your actual computer that you're typing on, but to Malibu, which is on the same network as the CHPC computers downtown. And so the file transfer is really fast. So we're going to now talk about this more. The last command is more genes.gtf. So you can type that command in, copy and paste it if you like, but I'm going to start talking about what this command actually does. So if I type more genes.gtf, or you can copy and paste it if you'd like, it is showing the first little bit of the file. It's more than 10 lines. I don't There's this kind of progress bar. It's saying it's showing 0% of the file. It's the very beginning of the file. So what you can do with more, this allows you to essentially scroll through a file. So when you type more genes.gtf, you get the first page. You can hit the space key and it'll keep paging. Each time you hit the space key, it will go to the next page. And page in this context is a little bit different. It's like the next n lines. Again, it's looking at that new line character to figure out which lines to grab and display. So you can also use the up and down. You can use the up and down characters, or you should be able to. Well, my keyboard isn't set up for that for some reason. Typically, you can use the up and down arrows. Oh no, that's actually the less command, which we're going to talk about in a second. The limitation of more is that you can only go forward. Okay. Now, one of the problems that everyone faces the first time they use the more command is that you don't know how to get out. So you're scrolling through, all right, I've taken a look at this. I'm good. Don't really need to look at it anymore. How do I get out of here? Well, you type Q, and that Q to quit and return to the prompt, that shortcut is used in a lot of different Unix commands. So now that I typed Q, it got me out of the genes.gtf file and gave me my prompt back. Okay, so in a nutshell, more is just to page through a file and do a little bit more than cat, in the sense that you can move on to the next step with a simple typing of the space key, rather than like, typing a new command in to look at the next 20 lines and the next 20 lines. All right. So the old adage is that less is more. So the inventors of commands for Unix thought it would be really funny to create another command called less, which is a little more sophisticated than more. Okay, it does the same thing in concept. So I can do less G tab to autocomplete, and I get the same sort of setup where I got the first page. I can hit the space key lots of times and just scroll through this guy. But it's better in another sense, because I'm now hitting the up arrow over and over to scroll back one bit by bit. And then I can hit the down arrow to move forward again. So this allows you to do like what you would do in a normal text editor like Word. You can scroll up and down and move and navigate throughout the file. Therefore, there's really not any reason to ever use more. You should always use less, but I introduce it just so you know where less came from, and you can know the history. The other cool thing in less, and I think this is, now we're getting into something that you would actually do quite a bit. All right, so you're working as a first-year graduate student studying muscle development and gene regulation. Your lab probably has some candidate genes that they're interested in, or maybe your hypothesis is that, things that drive aberrations in muscle development are probably protein coding. So you want to know, as this example shows, you might want to know where all the exons are in this, where all the annotations for exons in this file are. Okay, so I'm going to hit Q to get out of the less command, and I'm going to show you how we do that. It's if you get lost, refer to the text here. It's telling you exactly what to do, but I'm going to go through it. If I type less genes.gtf on the command line again and hit enter, what I'm going to do is hit the / key, and this for me is I think on all keyboards is the key that also has the question mark on it. So if I hit /, that's telling less that, okay, I'm about to search for something. That's the search interface. It's like hitting Control F in Word. I'm going to type exon, and then I'm going to hit enter. And as you can see, it highlights what I can do is I can type N for next, and it will scroll through. It will scroll through each of the occurrences of exon. Okay. So you can then search for another term like, well, I'm going to look for a gene that I like. I'm going to look for genes that are potassium channels. So K for potassium, C for channel, Q, I don't remember why. And then I'm going to hit enter, and it's going to work for a little bit. It's thinking. This is a massive file. So it's going to search and search and search, and it might come up empty, but we'll see. It's calculating the line numbers, and now it finally found some gene names that have KCQ in them. I don't recognize these genes. This isn't quite what I wanted, but it demonstrates the point. You can search the entire file for the first occurrence of the pattern you're looking for. So this would allow you to, search for the gene of interest. Okay. Given audio problems, I just want to check in. How are we doing? Everyone with me? Anyone not with me, lost, or can't hear anything, which is a...
You're doing okay. You pause, but it seems to not drop any words this time. So it looks like it's doing better.
Okay, good. All right, well, hopefully this will be the last time that happens. Okay. So one of the problems with less, as you can see, is that there's this line wrapping issue. This entry right here is a really wide line in the file describing lots of different things about this apparent exon associated with gene PRKCQ. But my screen is only so wide. So what UNIX does by default, or less in this case does by default, is it takes that really long file or long line and wraps it around so that it all fits in the screen space that we have. But that's good in some contexts, but it's bad in the sense that you can't really see which parts of the line correspond to the particular columns that are in this file. So more specifically, the GTF format has a column. The first column is the chromosome on which this exon lies. The second column is the data provider. So in this case, there's a group in the UK called Ensemble. They're the ones that created this GTF file with all these gene annotations. There's a third column that tells us what type of sequence annotation this is. So CDS stands for coding sequence. And these are the genomic coordinates of that exon. So this exon exists on chromosome 10 from that position to that position on chromosome 10, and so on and so forth. And the most problematic column in this file is this massive one here, which is just a concatenation of a bunch of information. And that thing goes on for, looks like a couple hundred characters. And so essentially that column is causing this to wrap around. So what I want to show you is you can use the less -s option, which is for scroll left and right, to basically prevent the line wrapping. As a consequence of preventing the line wrapping, we only get to see a little bit of each line. It stops at the beginning of that big, awful glob of characters I was talking about. But we can use the right key, the right arrow key, to scroll through and see what's over there. And now I'm using left to go back. So if you do less-S on this file, you should be able to use your left and right characters to just play around, and you can use the down character to move to new lines. So you basically have the ability to, by analogy to Microsoft Word, use the scroll bar on the right and the scroll bar on the bottom to move left and right or up or down respectively. Maybe I got that backwards. Okay, so again, last time, we're going to hit Q to get out of this and get back to our prompt. Right. All right, so in the last 40 minutes, search for patterns and files in a more effective way, because often what we're doing with genomic files is actually searching. They're huge, they encompass the whole genome. The human genome's massive. Typically we want to find subsets of the data therein that are relevant to what we're doing. So that inevitably boils down to searching. Okay, so we'll get to that. Quick tangent is to introduce another command called man, which is short for manual. Every command in UNIX has a manual associated with it. So there are, if you look, this is the manual page for the less command. There are boatloads of options. The only one I showed you was -S. There is a -A, capital B. I don't know what any of these do. I've never really played with them at all. But if you wanted to, master the less command, if you were super bored one night, you could type man less, and you'd get this man page. And what's happening, the man page is itself a document that is loaded into the less command. So there's this sort of meta-ness here, but so now I can use the arrow key to scroll up and down through the manual page, and I can use right and left to scroll through. Okay. There's not, I don't know of many occasions to do anything much fancier with less than what I showed you, but you might run into a situation, or you forgot what I told you with a capital S. Use the man page to scroll through and read it. I mean, reading this is laborious and boring, but all the information you might want is there. Okay. So if we wanted to answer this question before, how do we get rid of this wrapping behavior, and we want to use the manual command, what we did eventually find is that the less-s command, which I'll show you here what the documentation looks like. Now this is a bit hard because finding it is challenging. Well, now I'm not even sure I can find it. There we go. Causes lines longer than screen width to be chopped rather than wrapped. And that allows you to scroll left and right. Okay. All right. So now we're going to talk about the GFF format or the GTF format. All right. So nope, I got it wrong. GTF stands for General Transfer Format. The G doesn't even stand for gene. That kind of blows my mind. And the GFF format stands for General Feature Format. I'm gonna make this a little bit bigger. But this is a 9-column file. And as I mentioned, the first column is chromosome, then there's the source, and then there's a feature like it's CDS or exon or intron or whatever. There's start coordinates. There's a score column, which tells you sometimes the confidence in the annotation. Maybe it was predicted by a computer program. So there's some exons that are highly confident, some that are less. There's a strand column, plus for that gene is on the same strand as the reference genome. Negative if the transcription is on the opposite strand of the reference genome. There's a coding frame. So obviously you can, there's 6 coding frames, 6 possible coding frames, and then starting in the first position, second position, or third position on the positive strand or plus strand, and 3 coding frames on the reading frames on the opposite strand. So this will give you some information about that. And then the 9th column is this big hideous mess that I talked about, which encapsulates all this information about the annotation. No need to memorize the content, like what the columns mean. You can always refer back to documentation for this. But the point of this at this point in the class is to tell you that formats like GFF and GTF, they, just like this hidden /n character distinguishes rows or lines in the file, UNIX likes files, the columns in files, text files, to be separated by another hidden character, which is a versus /n for new line. So when let's be concrete and show genes.gtf. I'm gonna make this big. So this character, this big space here between 1 and Havana, is not someone hitting the space bar 5 times or something to make that gap. It's actually typing 1 character. It's only 1 byte, and that is by hitting the tab key. And that spaces out between 1 and the word Havana. So in Unix, if you want to ask, and I'll show you how to do this in a little bit, but if you try to ask the computer with a specific command to print just the second column of a file, what it's looking for is for each row, for each line, it's looking for a tab character. After the first tab character that it encounters, it interprets the first non-tab character after it to represent the start of the data in the second column. It defines the data in the second column from that point to the point at which it encounters another tab column. Third column, the same thing. It's gonna look for the second tab, and the data after the second tab is the third column, and so on and so forth. Okay. So sort of one of the main concepts of the lecture from today is that data files are represented, text files in Unix need to be represented in a very specific way. They need new lines at the end of lines, and they typically need tab characters to separate columns in your data. Now, fortunately, most of the file formats that you'll download in genomics follow that structure, but often you're going to be faced with the task of generating your own data files. So you need to make sure that those data files use new lines at the end, which with most programming languages or text editors, they would anyway. But where user intervention can go awry is a lot of times you would type, most humans might just put a space in between each column rather than a tab, and that will lead to problems. Commands like the ones I'm going to show you at the end of the lecture today won't work in the way you expect if columns aren't separated with tabs. Okay. Now I'm going to show you one other thing just quickly. Yeah. All right. So this is a ugly file, but just like cat-e will show that $sign to show us where the new line characters are. Cat, let me just do this. Head, I'm going to take the first 3 lines of the genes.gtf file, and I'm going to introduce a concept that I'll show you later, which is the pipe symbol. So this is the vertical bar. It's the character above the backslash key. On the backslash key, if you hit shift, you get the pipe. So this will take the first 3 lines. If I add pipe, it'll take those first 3 lines from the first command, the output of this first command, and pass them as input to the next command. So now I can take those first 3 lines as input to this command. And so it shows not only the new line characters for these first 3 lines, but if there were tabs, there's not, because we're not into the data lines yet, it would show a tab character. So I'm going to add more lines here. And what we see when we get to the first data line, this caret capital I between 1 and Havana, that is what's, for reasons that are unclear to me, the caret capital I is what's meant to represent that hidden tab character. So if you see $signs at the end of the lines and caret I's between data, that tells you you've got a properly formatted file. Okay. One command and passing it automatically as input to the next command. So this is our first example of being able to chain together, or so-called pipe together, multiple Unix commands, but that each do something simple, but in combination, they can do something quite sophisticated and powerful. All right, so back to the slides. Okay, So we're going to spend the next probably 20 minutes on this command called grep. Grep is one of the first commands that we encounter in Unix that doesn't have an obvious mnemonic. It's not clear what grep does by the word grep, at least not to me. I'll explain where it comes from in a second, but it is the search command. So this command here will search for the word exon, anything in between double quotes. It'll search for that pattern in this file called genes.gtf. So think of grep as search. It stands for general regular expressions and patterns. Regular expressions is a computer science concept, which is a way to define patterns to search for. But let's just run this command and see what it does. So it's just going to go and go and go and go and go because I've asked it to report all the occurrences of the word exon in this genes.gtf file, and that's what it's doing. You can probably see, depending upon bandwidth here, but you probably see it on your command. On mine, the word exon is highlighted in red. It might not be the case for you, depends upon how things were set up on Malibu, but I'm going to show you how to get that highlighting in just a second. But ultimately what this is doing is reporting all the lines, the subset of lines in this big file that have the word exon somewhere on the line. And again, the line is being defined by that new line character. This file is 1.5 gigabytes long. So this thing probably would scroll for the next hour, actually. All I wanted to show you is that the command is actually reporting things. So I want my prompt back. I want to move on. I don't want to stare at this for the next hour. So I'm going to hit Control C and it stops. Okay, I have my command back again, or my prompt back again. So how do you get the color? Well, the way to do that is I've set up with a trick that I'll teach you later. I've set up a trick so that anytime I type grep, it actually changes my command to be grep -- color behind the scenes. If you don't have that, if you didn't have that on a new machine, or if you don't have it now, you would just do grep -- color, double quotes, exon, double quotes. That's the pattern. That's the pattern between the quotes, and then genes.gtf. So now here's another example. Nope, I got a chat message here. Great question. So Jason asks, is there a reason why the color option requires 2 dashes versus the 1 dash that we've used before? Yes, it is a Unix convention. Any option that just has one character like -N or -A, you only use 1 dash before it. If it is a multi-character, multi-letter option, you have to use 2 dashes. That convention is violated by lots of software, including stuff I've written, but that is the general convention. Partly you just kind of memorize it more than anything. All right, so here's another trick. We've got grep -- color, and we're searching for exon. As we learned a second ago, this thing will just go on and on and on forever. I don't want to do that. Maybe I just want to do a small experiment to make sure that this is really matching the things that I want it to match. Well, if grep with the pattern exon, its job is to find only the lines in the file that have the word exon, I can test that by just piping pausing again. There we go. So this will just report the first 10 lines. The color went away though. The reason for that is that the color only exists for that first command. Once I pass off the results from that first command to head, that color bit is gone, unfortunately. But what we can see, but you could do this if you wanted it back. You could just put grep-color exon after the head command and you get it back. So here's an example where we find the subset of lines with exon. Head strips off that color information, just reports the first 10 lines that match that pattern. And then if we want the color back on, we can take those first 10 lines as output from the head command, hand them off to grep again and get the color back. So okay, so what did we do here? This is an example of testing our results, doing a small pilot experiment to make sure that the command that we issued actually is doing what we think it's doing. So I can't emphasize this enough. It's just like doing molecular biology or any other science. You have to sanity check your results. Check it multiple ways, multiple independent ways. Don't trust that just because the command is running, that it's actually doing what you want. Ronald Reagan is famous for something that a phrase that you've probably heard, but trust but verify. So always, trust that it's doing roughly what you want, but verify that it's precisely what you want. All right. So grep matches with color. We learned that. Now what I want to show you is a more sophisticated way of using grep. And one example of what we're focusing on here is that when I searched for exon, you can see that it caught, in each line, it caught this case, which is the word exon surrounded by white space. So so essentially that's a column in this GTF file with exactly and only the word exon in it. But later on in these same lines, we see that there's other instances of the word exon, but they're part of a larger word. Like this is exon underscore number, exon underscore ID. Ensemble even gives IDs to exons. And I guess this is the second version of the definition of this exon for this gene. There are many cases where you want to limit the lines that you match or you find with grep to solely cases where the word, just that word itself exists as a match. So if we wanted to search for gene, gene would, if without the -w command, would come up here, gene underscore version and gene underscore source. But if we use the -w, that stands for a word. It's essentially find this pattern gene, but find it as a word, meaning that it's flanked by white space on either side. There's nothing but the word gene, okay? Just like a word in a sentence, it's flanked by white space. So if we do that, take this whole command. And since we're using --color, we can see where that match is. And it's not matching. Now it's not matching this. It's not matching that. It's only matching this instance of the word gene in that third column, which is if you remember, you probably don't. But if you go back to here, that third column is the feature type. So this feature type on this line is essentially the definition of the entire gene. It's going to report the coordinates of the transcription start site of the sort of the longest isoform all the way down to the transcription end site of the longest isoform. Okay. Any questions about grep or -w so far? Everyone with me? Okay, great. So this is software or other programs. You can also ask if grep with the word gene with -w finds all the lines that have gene as a word somewhere in the line. This -v option does the inverse, V for inverse. It will find all the lines in the file that do not contain the word gene anywhere. Okay, so let's try that on this file. Guess what? The only lines that we get that do not have the word gene in them, do not match the pattern gene, are the first few lines, which are header lines defining what version of the human genome this comes from, what data was created, and some other stuff that we don't care about right now. Well, why is that? That's because all the data lines, this is a gene format. It's reporting all the genes in the human genome. So that pattern gene is on every single line because all the information, all the data information, all the data lines, sorry, in this file all contain the word gene. But let's do another example. Genes are comprised of exons and introns. So we could grep for the word exon, and let's do a quick example here. We use the -W option to find only the lines where exon is surrounded by white space. It looks like a word. So I'm just gonna take a peek at that. So it's cases like this. Now what we're gonna do is do the inverse of that. So we should not get any exons in our output. Let me clear the page so we get a good look at that. So the first few matches, yep, this one says gene, doesn't say exon on its own anywhere. This one has gene on its own. This one has transcript on its own. Okay? So you can use combinations of these kind of patterns to find exactly the type of thing that you want. So you can include patterns and you can exclude patterns by using or not using the -B option. Okay. So in this case, this is basically just what I did. I combined V and W to find report lines that lack a pattern, a specific word pattern. So we'll skip ahead. So now we're going to run into a common problem that basically everyone working with this file format runs into at some point. Let me show you results of this command. So a gene that's kind of famous in autoimmune disorders is PTPN22. It's especially implicated in type 1 diabetes and ulcerative colitis and other things like that. So it's the reason this is on this slide. It's the gene I worked on like 10 years ago. But what you notice about the format is, so when we search from PTPN22, there are lines that match exactly PTPN22, that gene, but there's also these alternative transcripts like -208, and we might want to just match on, might want to really restrict our matches to just PTPN22, but you'll notice that the format is kind of weird. The GTF format is notoriously, well, for lack of a better word, awful. It's not friendly for computers. It's friendly for humans to read, but not friendly for computers to read. A classic example of that is every time there's these pairs of information like gene name, and then after it will give information in quotes, then a semicolon, then the gene source, the associated gene source in quotes, semicolon, and so on and so forth. So the problem is, if we want to match exactly PTPN22, what really separates it, instead of grep asks us to put the pattern we're looking for. Let me say that again. grep wants us to put the pattern we're looking for in quotes. but the pattern we're looking for in this case also has quotes. So the way we handle this is with what's called an escape character. So this means that I want you to take literally the quote sign when you're matching. Don't interpret what's between the quotes as the pattern we're looking for. Include the quotes as part of the pattern. So you have to escape those quotes so that this is another example of where UNIX is sort of dumb. It's powerful, but it's dumb. It doesn't know exactly what you want it to do. You have to be explicit. So in this case, now we just get the matches with the quotes flanking it. In contrast, let me just show you. We not only get those matches, but we get these other matches where PTPN22 is followed by other junk. Okay, so in contrast, now we don't get these hits over here. We just get that because we've asked grep to take the quotes literally when it's matching, when it's searching for matches, include the quotes, in other words. Okay, so I've already touched upon this, but you know, if you've ever heard of a Rube Goldberg machine, which is what this video is about. Let me just take a break and show this because I love it. So you probably know who Rube Goldberg was. But he was notorious for these things, and that's where I want to stop. Where it's these ridiculously complicated contraptions that do very simple things at the end of the day, but they're just sort of hilarious for their complexity. And so sort of the output of one device would trigger the next device and so on and so forth. And that's what we're able to do with UNIX, where grep does something very simple. It just searches for patterns. Well, we know that there's going to be millions and millions of lines, maybe not millions, but at least, well, yeah, millions and millions of lines in this genes.gtf file that have the word gene in it. We can hand off those millions to this next command called head, as long as we use this pipe separator, and it will just report the 10 matches, the 10 first matches with gene in it. Similarly, we can figure out exactly how many lines have the word gene in it by piping the results to WC-L. And the -L option is a subcommand of WC, which just reports the count of lines. And again, it's using that new line character to do the counting. So apparently there are 2.895 million lines in this big GTF file that have the word gene in it. Now, I hope that example kind of demonstrates the power of how fast you can do analyses like that. Imagine if you opened up this hideously large file in Word. To answer the question we just asked, you'd have to open it up. Word would sort of spin its spinning wheel of death for like a minute until it opened the thing. And it'll start you at the beginning of the file, and you'd have to use your mouse, drag it all the way down to the bottom of the file to count how many lines there are. That's no fun. Moreover, it's not reproducible. There's manual intervention to do that. What's really nice about these kind of commands is you can, once you have a command that you feel like does the analysis you want, it will work forever. So you can document that command in the lab notebook. This is how you make your work reproducible. So this is an example of how you can do analyses quickly. This is much faster than doing it manually, powerfully and reproducibly. So now what we can do is get even fancier. We can sort we can take all the gene lines. Let's make it the ones with the word, the pattern gene is a word in that second column. We can sort the results, and then we can take the first few lines. Now, sort in this case is going to sort all the 2.895 million lines that have the word gene in it, and it's going to sort them first by it's then going to look at the next character. There's going to be a lot of ties because they're all Havana, and then it's going to go to the next column and break ties based upon alphabetical sorting of this first character in the next column, okay? Now, sorting 2.895 million lines takes a second, so that wasn't instant. But now what we get is, hey, wait a second, 10 is first. Well, the way that sorting works in UNIX is a little bit annoying, but 10 actually comes before 1 in this sorting criteria. We could fix that, but we could sort numerically. And we'll get more into this. Now this sorts in a totally different way, but at least the data are ordered. We'll get more into doing sorting in a better way, but you can see that sorting in different ways orders the matches in different criteria, in a different manner. Okay, so I just want to make sure this is really clear because this has become relevant in the homework, and really this is one of the most powerful things of UNIX is being able to chain together things with this pipe command. Whoops, what did I do? Wow, I'm not sure what I did here. Oh, I see what I did. Sorry guys, I inherited my son's gaming mouse and it has buttons on the side that I'm not used to. And I just clicked one. Right. So I've talked about this. It's probably clear. I'm gonna beat a dead horse here, but we take one command, it's handed off to the second command. That second command does its thing, spits out its results, hands it off to the third command, and so on and so forth. And then finally, if there's no pipe afterwards, the results of this Rube Goldberg-like pipeline are spit out to the screen. Okay. What I should emphasize as well is that there's no limit to the number of commands that you can chain together in that way. There's sort of a human limit in terms of our capacity to understand and predict what the final results will be if it gets sufficiently complicated, but there's no technical limit to know how many combinations you do. Okay, so just another example, we can take, oops. All right, well, let me just go back to PTPN. I'm going to use my trick here, Control R to find PTPN. So I got it. And then I'm going to pipe it to WC-L. And I get 316 lines. So I found all the matches with PTPN with quotes, part of the match. And then this is just a nice, I can write this number down in my lab notebook or and write down the command or save it in a document so that I have a command and it's all reproducible. And this command takes a little while. We have a faster machine, so it's not 20 or 30 seconds anymore, but it takes a little bit because it's literally starting at line 1 in that file asking, is there a match for PTPN 22? No. And then for millions of lines, it's probably answering no, 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 no. And then finally, it finds those lines that have PTPN 22 and prints them out. But it has to essentially scan the entire text file and test yes or no, is there a match on this line or this line or this line? Okay. So we can ask how many exon records there are for PTPN 22 by just piping that first grep for which finds PTPN 22, then grepping again for exon, and then counting there, same thing. So this is one of the key strengths. And then in the last couple minutes, what I want to introduce is the cut command. And cut so grep allows you to find lines that match, extract lines that are relevant. Cut allows you to extract out particular columns of a file. based upon that tab delimited character, that tab character. So it assumes, the cut command assumes that your file or input, the input coming from a pipe and as output from a previous command, is tab delimited. So let me show you an example of cut. I can say cut-f for field. I think this is unfortunate. Field is analogous to column. So if I want the second column, I'm asking cut for the second field. So if I do this, the second column is this data provider, which is ensemble. And what we get is it's going through every line of that file, but all it's printing to the screen is the data provider, the value of that second column. And it knows where that second column is because of the tab character. We can also ask for columns 2 through 4. The dash indicates through. So that would be 2, 3, and 4. And what you can see is there's one column, column 2, another column, column 3, column 4. I can change it to column 2 through 3. Or I could do 2, 3 if it's just, so I could do 2, 3, 8 if I wanted to skip columns 4, 5, 6, and 7. Okay, so this is, it may not be apparent yet, but this is really nice for subsetting your data in useful ways. And really what I'm trying to show here is that what's hidden behind the scenes here, denoted by these blue bars, is this invisible tab character. And that's how it defines what columns we actually extract with that -f option, okay? One maybe surprising thing is that if we look at the first 10 matches, it also includes this garbage at the header lines at the beginning. And I think that's kind of an idiosyncrasy of this command, but if it can't, if it cannot find any tab characters in the line at all, it just reports the whole line. I don't really like that, but it's something we have to live with. If we wanted to get rid of the header in our matches, we could use grep-v, because we know all these header lines start with a pound character. So what I could do is take the grep-v pound character. That's just going to give us the data lines. So I can pipe this to less and show you that. The first line is a data line. And then I can say cut-f2. Show you the results of that, the first 10 lines. And now the header lines are gone because we've stripped them off the file by grepping, hand that off to the cut command now that we have data lines, and now we're looking at the first 10 matches, okay? All right, I have 15 seconds, so I will stop. I'll make a note to pick up on these last few slides when we start on Tuesday. But what I want to just mention is the first homework is assigned today. It's a pretty straightforward homework, shouldn't be that hard. This is just kind of reinforcing the commands that you've learned already and getting your feet wet, playing with these commands on your own if you've been following along. As I mentioned in the first class, this will be due at 11:59 next Thursday, so a little over a week. And what you'll end up doing, what I want you to do, is make your answers to the homework in a text file that starts with your unit and is labeled homework1.txt. And what I'm going to do on Sunday is I'm going to post a Dropbox link for homework 1, where you'll just essentially, there'll be instructions on how to just post this homework file to that Dropbox link by 11:59 next Thursday. Okay, and I'll probably follow up on Slack about instructions there. But if you have any questions, follow up. Also, just want to let you know that we're going to which is a bit of a problem for talking back and forth. So they're gonna post new Zoom links on the GitHub site for TA hours starting. The first one will be tomorrow, and then there'll be one on Monday and Wednesday. So I apologize for the audio and video issues. I will henceforth teach from my office at work, and that should mitigate all this. Are there any questions at this point? Nope. Okay, great. Well, thanks so much for bearing with the technical difficulties, and we'll see you next Tuesday.
Thank
Bye.
you.